Hi Dentrism students, it's Dawn. I'm just wanting to finish up that small bit of information that will be on the test on Monday about the dental pulp. So we'll be looking at the, follow along with the slides that I provided you that we just ran out of time covering the other day. I hope you all had a great holiday too, by the way. Beautiful weather. All right, so we'll start with these. This is a cast, those were casts, like acrylic casts of actual pulp. So I'm just going to go back to that screen again so you can see if you were to take the pulp tissue and the shape of the pulps of each tooth in the mouth, this is what they would look like. So it kind of gives you an idea of the uniqueness of each tooth and its anatomy. So basically the pulp tissue develops as the tooth, the crown of the tooth and then the root of the tooth form. The cells that are in that dental papilla area are from the mesoderm originally. So some of these cells we talked about before will differentiate and become odontoblasts that will produce dentin. The rest of them stay in the center of that dental papilla area until the tooth forms around this structure, capturing any blood vessels, nerves, nerve endings, etc. that may become trapped in there as the root forms and and we'll talk more about the epithelial root sheath later on, but I've mentioned it before. It's going to get narrow as the root length forms and will end up being just a small opening at the apex of the root. The human dentition generally has 52 pulp organs. Of course, 32 of them are the adult dentition and 20 of the primary dentition. We have the pulps that are found in the molars. The pulp tissue is found about 4 times larger than the amount of tissue that's found in the anterior teeth, simply because of the size. The main function of pulp tissue is to provide support to the tooth, maintenance of the structure of the tooth, and that includes the continued formation of dentin. We'll find some principal cells, generally speaking. We've talked about that dental papilla being undifferentiated mesenchymal cells. There will remain a large number of these undifferentiated cells that can act as a cell pool and will differentiate when they're needed. Many of them will differentiate into odontoblasts or fibroblasts. They make up the largest types of cells that we find in the pulp. We also will find our macrophages and our immunocompetent cells. So when you start talking about inflammation and how the body deals with that, that's where macrophages and immunocompetent cells come in. So they're basically there to help get rid of any old dead cells, and they're very, very busy because there's a large turnover in the pulp. We'll look at the fibroblast first. It is the largest the most numerous cell that is found in the pulp. It's found throughout the pulp, and its function is to form and maintain a pulp matrix, which is the goo that holds all of the structure of the pulp together, called the ground substance, as well as fibroblasts have the ability to produce collagen. So this fibroblast cell can produce the ground substance that the structures float around in, as well as the collagen fibers that make up the fibrous component of the pulp. The fibroblast differentiates from that pool of mesenchymal cells. The unique thing about the fibroblast is when we talked about protein synthesis, the fibroblast will actually be able to produce, it is a blast cell, it makes collagen fibers, but it also, within the same cell structure, can break down and degrade collagen. And so therefore it's like acts like a classed cell within the same cellular component at the same time. The other cell that we see are the odontoblasts, and these are the dentin-producing cells. They completely line the walls of the pulp chamber and the canals. Other cells that are there are just undifferentiated cells that float and will differentiate when needed. Now the fibers in the ground substance. We have type 1 collagen fibers that are produced by the odontoblast, and then type 3 collagen fibers that are produced by the fibroblasts. So both odontoblasts and fibroblasts are capable of producing the fibers that contribute to that pulp material. And the fibroblasts also produce the ground substance, which is made up of a number of proteins like glycosaminoglycans, proteoglycans, glycoproteins, as well as water. There are 6 main functions of the pulp. Sorry, 5 main functions of the pulp. The first one we've talked about as far as the initiative function. It's the cells that are found in the dental papilla originally that have the neural crest cells influencing them. It's those cells that actually cause the tooth to start to form. It tells the enamel organ to start getting ready to form a tooth. Then the cells that are found in the dental papilla, some of them will differentiate into odontoblasts that will have a formative function for producing dentin. Should that tooth ever be stimulated by some trauma of any kind, it has a protective function. The cells that produced dentin, the odontoblasts, can produce reparative dentin. The fourth one is the nutritive function of the pulp. It brings, because of the presence of the blood vessels and the veins going in and out through the apical foramen, it carries oxygen and nutrients to the developing and once functioning tooth. And then the reparative function, again, it's very similar to the protective, but if there is a need for the dentin to repair itself, it will, and that will protect those precious cells of the pulp. Now the pulp anatomy, basically there's 2 components to the pulp. We have the coronal pulp, which follows the contour of the crown. It has usually 6 surfaces, including the floor. And then the radicular pulp, which is the singular or seen in singular or multiple teeth roots, and they're tapered, running down the roots to the apex. And so all of the nerves and blood vessels enter and exit through the apical foramen. Now we'll be looking at all of the different zones of the pulp. This is a cross-section of a tooth that is lined with odontoblasts. And if we look at it close up, you'll see these are the odontoblasts that line the pulp. Next to that is the cell-free zone of whale, which is right beneath the odontoblasts. Mainly we see this in the coronal pulp. Next to that is a cell-rich zone, and this is where there's a multitude of different kinds of cells, like our fibroblasts, our undifferentiated mesenchymal cells, our immunocompetent cells. Those are all there to help replace anything as needed. And then you can see the fibers that are in this diagram as well. And then below that, we have the pulp core, which is where the blood vessels and lymphatic nerves, as well as more fibroblasts, are found. So the pulp tissue is highly vascularized. There are thin-walled arteries that enter and exit through the apex. They're either myelinated or non-myelinated neurons found in the pulp that will terminate in and around the odontoblasts. Their main function is for pain reception. The maxillary teeth are innervated by the maxillary division of the trigeminal nerve. The mandibular teeth are innervated by the mandibular division of the trigeminal nerve. So in summary of the pulp, we have these thin-walled blood vessels, nerves and nerve endings, cells like fibroblasts, odontoblasts, undifferentiated mesenchymal cells that are all floating in a supportive ground substance made up of glycosaminoglycan and collagen fibers. Now, one of the most interesting things that we see and the most complex things is the pain-pulp dentin complex. There are three hypotheses of dentin sensitivity. I don't know if you've ever had any sensitivity of the teeth, but it can be brutal. It's very difficult to cope with. There are lots of products that are on the market that will help with this, and it just helps to understand how it's happening. The three hypotheses of dentin sensitivity are the direct innervation theory, the transduction theory, and the hydrodynamic theory. And I'll briefly go over what each of those theories are and what the most common one is, so it helps understand how we treat it. The first one is the belief that the nerves that are found in the pulp core actually will extend between the odontoblast as far into the tooth as to the dental-enamel junction. However, they've done numerous studies and they have proven that there are no nerves found at the dental-enamel junction. So this is not a very commonly accepted theory. The second theory is rather interesting because it has to do with the fact that the neural crest cells, which are, remember, they were the cells that formed the neurons in the spinal cord in the brain. When they migrated down into the facial area, there is the theory that the odontoblast, because it was differentiated from that interaction, will actually act like a nerve, and it acts as a receptor and can conduct and contract that pain message to the nerve endings that are then sitting close to the pulp. This has not been proven. It's not likely to be the case. They haven't been able to show any kind of a synapse or any way that that message is passed on through the odontoblast cell. That leaves the third hydrodynamic theory is actually the most plausible and accepted theory of dentin sensitivity. And this is when we look at the tubules of dentin, once they are exposed, let's say from toothbrushing away enamel at the cervical area or fractures or anything like that, those tubules are now little holes, and fluid can be pushed into the tubule, causing that process that extends from the cell body, the odontoblastic process, to move against the nerve endings in the periphery of the pulp. So we've got that blood supply and nerve supply sitting in the center of the pulp. And then as that cell, the odontoblast, gets compressed, it puts pressure on and deforms the nerve endings that are sitting down in the pulp area, which then in turn acts as a mechanoreceptor and produces an impulse that is then sent to the brain saying, ouch, that hurt. So this is basically what the hydrodynamic theory looks like. The fluid open tubule on the ends leads these little pores, and it causes a fluid flow in here, causing the odontoblastic process to shrink, affecting that cell body and the nerve endings that are situated in the pulp that cause reaction then sent to the brain. So the products that we recommend we use, such as Sensodyne, is a product that is put on the surface of the tooth to pack those tubules. It has the ability to penetrate into the tubules and seal that up. Some products are more effective than others, and it's just a matter of trial and error, basically. The last thing I wanted to mention about the pulp is that over time, there are regressive changes that will occur in the pulp. The number of collagen fibers is increased, causing the pulp chamber to be fewer blood vessels. And then there's even little stones that will form in there called pulp stones. So I hope you have had a chance to take a look at these units now and prepare for the test that is on Monday. It's the same test that I provided you with before. And I hope you enjoy the rest of your week. And I thank you for your time.